Okay, thank you. Thank you for the introduction. And this talk is about how to predict the prevalence of complex genetic diseases using capsule networks. My first authors were Shao and Zhongbin, who are not here, they're in China. So I'm here doing this. And here in the title slide, you already see a capsule network, which is a capsule network that we ran. I'm going to explain in more detail what it's supposed to do. Yeah? So here, an introduction, we're focusing most of all on amyotrophic lateral sclerosis, ALS in short, but we can apply our method also for any other complex disease you want. ALS is particularly interesting because it is particularly complex. Yeah? So what is this? Actually, yesterday when I practiced the talk, I realized this is the exact same slides I ran 4 years ago when I gave a talk here in the VarI, and which felt wrong to me in a way. But then I feels a lot better today because I now catch up in terms of schedules. Yeah. So, and also I promise I have some new slides, so please stay. So here, this is the motor neuron disease. It degenerates upper and lower motor neurons in muscles weaken and shrink. And a prominent patient was Stephen Hawking. Everyone knows him, but he was very lucky because that usually occurs 2 to 5 years after first symptoms. And the two last statistics mean, okay, many people are affected, but you hardly see anyone. And why is this? Because of upon occurrence of first symptoms, the people just die quickly. And so, in other words, ALS has a very poor prognosis. Yeah? So, and why is this? As of the most current estimates, it's like 80% of the heritability is still missing. Yeah? So it's really hardly understood this disease in terms of the genetic background. It provedly has. Yeah? So we need to work harder for getting there. And the reasons usually are the usual reasons. Yeah, we have like epistasis, non-additive relationships that establish the disease phenotype. Yeah, on the one hand, and also, which is in the debate most recently a lot, is also the omnigenic model, which means due to the small world property, we have the core genes which are mostly responsible for the disease to occur. But then on the other hand, we have like small world property, like all of the genes being only a few steps away from these core genes, so have an influence on these core genes. So possibly every gene has an influence in this disease. And we would like to hook up to the idea here as well. So ALS has a particularly complex genetic architecture. I mentioned this at the beginning, and this is why it's so, as a disease to examine, so intriguing. Yeah? So a formal description, genetic architecture, for my purposes, this is, first of all, to have everyone on board. We have once an individual genome consists of 2 copies, and we look at the polymorphic sites, which are the sites where variation occurs, and we note down whether the parents agreed on the reference allele, whether they disagreed. This is a 1, 0 for reference, and 2 for the parents agreeing on the alternative allele. And then we have we have a vector of length approximately 3 to 5 million entries, and we have 0s, 1s, and 2s in this vector, and this is the input to our models. And so I frame the whole thing as X being all the people, and they're represented by the genotypes, yeah, and the genetic architecture F sub ALS is a function that maps one's genotype profile to either 0 if the individual is not affected by the disease, or 1 if it is. Huh? So that's the function we'd like to understand, and nobody has understood so far. And it's really difficult for ALS to understand this. Huh? We'd like to machine learn that architecture, which is we would like to learn that function using machine learning. Yeah? And so what does that mean? We pick a class of machine learning compatible functions, for example, a certain type of neural networks or linear functions, yeah, or support vector machines, whatever you may wish to use. Here we focusing on neural networks, but yeah, okay. So what you would like to do then is we would like to approximate that function which we do not understand by this function from this class of functions, and we'd like to figure out what is the best function from that class of functions in terms of the approximation working well. So, and here we use known, this is all standard stuff now. Yeah. So here using known examples, we have cases, which is individual genotype profile being labeled with 1 because they're affected by ALS, and the other controls. Yeah. So the goodness of the approximation is eventually evaluated on previously unseen test data. And so what you have to keep in mind here is that the data is expensive. So training validation test data reflect case and control cohorts. It's not that easy. Yeah. I mean, we all know to get one's individual genotype profile. So this costs, it's expensive. And so data, yeah, be sustainable with it. Yeah? All right, has this been tried before? Just machine learning the genetic architecture the way I framed it? Yes, if you want. Yeah, yes and no. Yeah, so yes, I mean, you can view genome-wide association studies, GWAS, as an attempt to approximate the true function with a linear function. Yeah, the advantage, which I don't object at all, yeah, is kind of the approximation is easy to perceive. You just look up what happened in the linear function. You look at the different entries, the different variables, and the coefficients going with them, the weights, and then you pick the ones that were the most strikingly, obviously, yeah, coming up. Yeah, and then, however, and this is the problem here with this disease, is we cannot capture non-additive effects with this, and so we need to do something else, which is not that easy to perceive. Yeah? And then this is what I presented 4 years ago, ALSNet, yeah, with some co-authors. A partial no, not really yes, because ALSNet was kind of proof of concept stuff, yeah, and it was the first attempt ever to do this. It does not provide a full answer. It left one with many open questions. So this is what it looked like. We had this huge black box thing here, yeah, deep CNN, yeah, and it achieved good accuracy on 4 chromosomes, did never work on the whole genome, and so, well, got confused when combining effects possibly, and so on and so forth, and we were left with some impractical choices, for example, pre-selecting chromosomes, stuff like that, yeah, and no further insight into disease mechanisms. So there were some things to be improved. Huh? So we using capsule networks here now. It's like, what's the purpose of them? The idea is here, this is a phase, yeah? Well, this is not a phase, yeah? And the CNN would classify this as a phase. The CNN has neurons that process scalar values, yeah, real values, yeah? And capsule networks, on the contrary, so the CNN would see, okay, so it's the contour of the face. Yeah, there's 2 eyes, there's a nose and mouth, all with high probability, the values in the neurons reflecting some kind of probability, and they're saying in the overall, the CNN concludes the face is there. Yeah, so and this is like classified as a face. The capsule network has vector-style neurons that do not only collect the probabilities into the vector, but also other information by which the capsule network can better classify what's going on. Yeah, so for example, here in this example, we also store the size and the orientation of the different elements, and then you see, well, the mouth is slanted, so the angle does not agree with the other elements, and also the size of the left eye is larger than the one the right eye. So the CapsNet concludes this is not a face. Yeah. So also they point out natural ways to interpret results. So this breaks open a little bit the black box character of many neural networks, and we require sufficient less training data. So this was good motivation for use them in a biological setting. Yeah. So we are hoping for mapping hierarchical structures and not being confused by this. Complexes, pathways, and so on. Yeah, CapsNet proved to be particularly well also in images when there were distorted or overlapping parts, and they could like disentangle that. Yeah, and here we're hoping not to be confused by overlapping interfering pathways. I mean, we cannot prove that it worked, but I mean, this was the hope, the motivation. Yeah, so less training data means less expenses. Yeah. This is the workflow. Yeah, all kind of real GWAS normal stuff. We clean the data and so on, remove all the artifacts and blah, 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 batch effects, yeah, population bar, and so on. And then in the end, we have gene PCA as a novel protocol. We reduce the dimensionality of the data, which is 3 to 5 million, yeah, the vector And now we apply not global PCA, which would linearize the entire data and not being applicable for any non-additive approach afterwards. So this has to be watched for. And we developed gene PCA, which is a local protocol. And then we preserve to be able to discover nonlinearity across the genes at least. Yeah. All right. So this is the network. It's pretty much adopted from the seminal work. The input then is a vector of length 75,484, where the entries correspond to the gene principal components. I'm sorry, according to what I just described, and the output is just 0, 1. Yeah, ALS or not, or the other way around. Yeah. Results are disease capsule actually outperformed everyone else in terms of accuracy, raised accuracy from 77% achieved by LSNet to 87%, running on whole genome, and they're also very balanced in terms of its measures, in terms of the measures, precision recall, I mean. So for example, the best competitor here has no recall, so does not really recover the cases much, and ours is in many ways the best approach. It's also the most stable one across different runs of initialization and so on and so forth. Yeah. All right. So then also this is about the interpretability. So we discovered 922 genes that were decisive for the classification process to work. And so we studied the capsules, and we discovered one capsule firing the most in that thing. And the good thing here with the capsule networks is you can really go into the capsules and look, because you have a wholly different set of parameters, the so-called coupling coefficients, which do not apply for normal networks. And you can study those, and you can then more easily detect or discover the activation patterns. And so we found this primary capsule 5, yeah, collecting 922 genes that cooperated to establish the correct classification of ALS. Yeah. Okay, so you may want to look into those. We found that the Go terms that we could associate with them, they made sense. Yeah, like here, many annotations related with the nervous system. In conclusion, we have the feeling, yeah, capsule networks are a better approach to really, when you really would like to interpret results a little better. Yeah. Okay, then also what I was most thrilled about was this year. Yeah, it's kind of, we discovered 644 non-additive genes. Yeah, where what we did is, briefly, we ran a genetic algorithm. We determined a selection of genes that did not work at all in a linear scheme. Yeah, so the constraint was they are not supposed to work in a linear scheme, and on the other hand, they were supposed to be as well as possible in our nonlinear approach. And then we selected those. Hard work, genetic algorithm took us a long time, and nevertheless, we found these 644 genes to, well, actually a local optimum, but nevertheless, they are optimal. And we retrained the linear approach to give the linear approach really any chance it deserved, and it could not do anything but raising accuracy a little bit above baseline. Yeah, and we performed fairly well with 71.2% here. Yeah, okay. So there must be epistasis here. Yeah, this is kind of a proof. Huh? Okay, disease capsule also needed less training data. The performance did not collapse like the performance of all other approaches when decreasing the amount of training data provided. All other machine learning approaches, nonlinear approaches, the only one that did not collapse was the linear approach. Yeah. All right. So acknowledgements go to Project Mine, from which we retrieved the data, which is a great project. To my understanding, they certainly make progress in terms of unraveling this disease. So my acknowledgements go to them. Also to Jan Felding, for example, who is a PI of the project, who made sure that we got the data. My first authors, I mentioned them already, and the ALS Foundation, which provided money for supervising Lizam here, who provided a lot of input, and Marlene is, we're supervising her together. And Sumanta was a capsule network wizard in my group. And that's the reference. Thank you very much.
Many thanks, Alex. Questions?
Hi, I got a question. So have you looked at LD? Because another potential explanation for what appears to be epistasis is unmodeled LD. So, for instance, if you tried polygenic score generation methods like LD-PRED or PRSTS, which model LD, and if you compared your method against that, I'm wondering if you...
No, I actually never heard the name. Can you pronounce this like a little?
LD-PRED.
LD?
LD-PRED. And
LD. Okay.
So it's a polygenic score generation method which models linkage disequilibrium.
Okay.
And because linkage disequilibrium, if you don't model it, it can look like epistasis. So that's a potential explanation for why it looks like that you have.
Is this new or is this?
It's been around like 6 years now.
6 years. Okay. Okay. No, I didn't come across that. Happy to try. Really.
One more.
One more. Okay.
Hi, thank you so much. I think I was wondering whether doing some pre-training on non-ALS patients, either a general cohort like UK Biobank or 1,000 Genomes, or even other disease cohorts, might enable the method to learn some general principles of how interactions in the genome work, and then doing some fine-tuning with the ALS cohort, which is smaller.
I didn't have an Outlook slide. This is exactly the Outlook I would have given.
Okay.
Great question.
Let's talk more about that.
Yeah.